So hi everyone, thanks for the invitation. It's truly an honor to speak to you all today. And I'm so grateful to be given this opportunity, really. I feel like I moved out of India and I sort of lost touch with the progress that's happened there since then. And so it's amazing to see all of you and look at all of your work on all the amazing stuff that you do. And I hope you find this story interesting also. And so, as I said, feel free to stop me at any time if something isn't clear. A lot of this right now, I'm a postdoctoral fellow at NYU, but a lot of this work was done when I was a PhD student at UCSD. And so, sorry, let me just move things away. And so, in general, I am passionate about memory and understanding how the brain forms memories and how we retain those memories and how we call upon them whenever we need and whenever it's relevant. And I think like the ability to form memories is what defines us and what actually makes us and drives our personalities. And so before I begin, I just wanted to show you a clip from a movie that I'm very fond of. It's from Ratatouille. And the guy over here in the scene is a food critic who has a taste of this plate that's brought out to him, and you'll see in an instant what happens. And so the idea that I'm trying to convey over here, that memories have this ability to transport us to a different place and a different time in our lives. And if you think back on any of your fondest memories or times when you've been transported, you'll remember that not only do you remember all the details and how that made you feel, but usually always you link that to the certain space where you were sitting or where you were or what room you were or like what childhood place you were in. And so the idea is that actually, if you think about where memory and space is located in the brain, they're actually both in the same region called the medial temporal lobe, which is shown over here. And actually, the neurons that code for memories and space are overlapping, and they both lie in this brain area that I'll go into further detail soon. And so if we split apart what exactly and where exactly do the neurons in this medial temporal lobe area belong to, we can divide them into this area called the entorhinal cortex and the hippocampus. And there are other brain regions too that I won't talk about today, but they all coexist within this medial temporal lobe. And around in 1970s, actually, it was first discovered that these neurons in the hippocampus specifically have this unique property of firing for spatial locations. So they have tuning curves, or they fire whenever you're moving around in a certain location in space, and I'll come to that. And similar properties are also observed in the entorhinal cortex. And these series of discoveries were together awarded the Nobel Prize in 2014. And so the idea is that neurons within these two brain regions sort of form a GPS inside your brain to tell you where exactly you are at any point in time. And so as an example, I'm going to show you a video of what we call our place cells in the hippocampus. So these cells fire only when you're in a certain location in space. And so what we've done over here is, well, not I, but what is going on over here is we're recording from a rat as he's running around in this open box. In gray is his path, and in red is the spike of a single neuron. So anytime this neuron is firing, it's overlaid over here in this graph as a red dot. And what quickly becomes apparent is that this neuron chooses to more likely than not fire whenever the animal is in this area of the box. And so we can plot this as a heat map, but effectively what it tells you is that this cell chooses to fire anytime the animal is in this lower southwest corner of the box. And so because of this property, these cells are called place cells because they tend to fire in a certain place. And you can imagine that the hippocampus has millions of neurons or thousands of neurons that are firing in specific locations around space as you're moving around. And so this is supposed to be a sort of internal GPS telling you exactly where you are. In addition to these place cells in the hippocampus, we also have even more interesting cells in the entorhinal cortex, and these are called grid cells. And over here, it's a similar idea as the previous video. This rat is running around in the environment. His path isn't overlaid here, but again, the spikes of a single neuron are shown in white. And in the beginning, it looks like you can't really make out, the cell doesn't really seem to fire in any specific location. But as the rat starts running more and covering more of the area, you realize that these cells are doing something really incredible. They're firing in this perfectly hexagonal pattern. And that because of that, they're called grid cells. So it's almost like they're forming a 2D, like a grid of the space, almost like Cartesian coordinates on which you can then build a map on top. And because of these discoveries, these cells were awarded the Nobel Prize because they're really supposed to represent a Cartesian coordinate of the external world within the brain. And so these spatial tuning properties are really exciting because they give us an idea for how the brain computes location within from the external to the internal world. But sometimes these spatial tuning properties aren't enough. And so what I mean by that is, let's assume in this toy example that this rat is moving from left to right on this track. And you've got 5 example cells that have tuning curves or like spatial fields or place fields, as we'll call it later. Firing over here for the red cell, then the yellow, then the blue, then the purple, then the green. And so this sort of spatial overlap between cells can provide enough resolution for the animal to know when he's at this black location versus this black location. But theoretically, there is a limit to the resolution in which you can get. So for example, if the rat were to disambiguate between these 2 locations, it would become really hard for it to know, just purely based on the spatial tuning curves alone. And so this is where oscillations actually provide a solution to this resolution problem, in addition to several other factors. And so what do I mean by oscillations? I'm sure you guys are really familiar with EEGs and MEGs and probably also LFTs. But so when you record from the surface of the skull, you see low amplitude changes in the neural signal, which is the EEG signal. But the moment you penetrate inside the skull, you see these beautiful large fluctuation oscillations. And so this is just basically a linear probe that's implanted into the hippocampus. And you see these amazing fluctuations of voltages that are going on within the hippocampus. And in fact, these are found not just in the hippocampus, they're found everywhere in the brain. And these LFP measures, they tend to measure the synchrony of neurons or the synchrony of inputs to a certain brain region. And they're key for diagnostics for several diseases, as well as people think that they could potentially also be used for therapies for several diseases. And so specifically in the hippocampus, there are these theta oscillations, which is a prominent 6 to 9 hertz oscillation within the local field potential. And they're thought to be generated by neurons in this area called the medial septal area. And I'll get into the medial septal area in a little bit, but for now, all you need to know is that there are these oscillations within the hippocampus, which are really prominent around the 8 hertz range. And so if you remember, I told you that these oscillations provide a solution for this resolution problem. And so how do they do that? So what I showed you right now is that there's an ongoing oscillation in the hippocampus. And in addition, there are cells that have spatially tuned fields that tell you the precise location of where you are. But one additional property of these cells is that in addition to their spatial tuning, they actually also fire at a resonant frequency. So they themselves tend to oscillate. And if you're not clear what I mean by cell oscillating, just let me know. But basically, the action potentials fire in bursts, and these bursts are spaced by a certain time point, which is the frequency of that cell's oscillation. And so they tend to oscillate at a frequency that's slightly faster than the ongoing field potential. So what that gives you basically is that, for example, let's say this mouse enters this red cell's place field. And a second quality is that anytime the cell fires its first action potential, it'll always fire at the trough of the local field potential. So what happens is the cell first fires at the trough of the local field potential. And because it's firing at a frequency that's faster than the ongoing oscillation, the phase at which it fires keeps advancing by a little bit. And this is called phase precession, because the cell was earlier firing at this descending phase, and now it moves to the middle, and then now it moves to the ascending phase of this ongoing oscillation. So this is for the red cell. The same thing happens for the yellow cell. It fires at the descending phase, then the middle, then the ascending phase. Same for the blue cell, same for the pink cell, and then same for the green. And maybe you've already discovered what I'm pointing towards over here, but what this gives you is basically within the time scale of an oscillation, the exact sequence at which the cells have occurred becomes replayed within this time scale of a few milliseconds. So this allows perfect temporal resolution of your past trajectories to allow for things like plasticity and to allow for several other things that I won't get into for memory consolidation. And so basically the point that I'm trying to make is that not only is the spatial tuning of these cells important, but also their temporal firing is important, because this determines the precision with which they fire in relation to each other, given the trajectories that you've taken in the past. And so what I've told you now is that within the hippocampus, you've got basically two oscillations. One is your ongoing local field potential, and the other one is the cellular oscillation, which tends to be at a frequency slightly faster than your ongoing LFP. And so one question that I'd like to address in this talk is, how do these ongoing oscillations, the field potentials, influence the second oscillation of the neurons? Are they coupled? Are they completely independent? What's the relation between these two? And secondly, do these oscillations even matter? And this is a really tough question to answer. In fact, there's like these huge fractions of groups of people in the neuroscience community, one half that think oscillations don't matter at all, and that they're just epiphenomenon, and then the other that say, no, this temporal timing is really important. And it's been a difficult question to answer, because any manipulation that has tried to change specifically the oscillation tends to change something else too. You change the firing rates of cells, or you lesion a certain brain area. So it's really hard to say for sure that your manipulation causes changes only on the oscillation and nothing else. And that's what's driving your behavioral effects. Okay, so what I've told you so far is that neurons in the temporal lobe, they display spatially precise as well as oscillatory precise firing. In addition, what I didn't show you in detail, but we know that the medial temporal lobe is essential for generating new memories and for navigation. And so the question is, are the two connected? Like, do oscillations play a role in the function of the temporal lobe? And so to do that, I'll go through a couple of the aims and the short-term goals that our project tried to address. Are there any questions specifically about like any of the background that I covered so far? If not, then I'll proceed with the aims. But if something's not clear, then just let me know if like something about it. These are very hard topics to explain, so I'm hoping I'm doing a decent job. So the first goal that we tried to do is to gain control over these theta oscillations in a way in which we do not impact drastically other properties. And so we can truly answer if the oscillation itself is important without impacting many other downstream targets. And so then we can't make any conclusions. After that, we can then, if we can successfully do this, then we can address how spatial firing or these tuning curves are affected by changing these oscillations. We can also address how the temporal firing is affected by changing these oscillations. And then we can maybe address whether these oscillations are relevant for behavior and memory. And so we can talk about the first part where we're actually gaining control over theta oscillations. And so what I told you is that these oscillations actually arise from this other brain region called the medial septum. And neurons over there, specifically these GABAergic neurons, are thought to act as pacemakers for this local field potential. So they also fire at 8 hertz or whatever the frequency of ongoing LFP is. And their firing is what sets the local field potential within the hippocampus and the entorhinal cortex. And so the idea is if we can gain control of these neurons and change their firing frequency, maybe we can change theta frequency. And so over here we use optogenetics, which I'm not sure how familiar you are with it, but basically it's a tool that allows us to, it's an ion channel. And so with blue light, we can artificially control the activity of these cells. And so what we basically can do is express channel rhodopsin, which is this opsin, in GABAergic cells within the medial septum. And we can shine blue light over these cells. And every time the blue light turns on, basically all of these cells are forced to fire an action potential. And then we can record activity in the hippocampus and the entorhinal cortex and see what's happening when we do these manipulations. And so I express channel rhodopsin in the medial septum. These are like different sections covering the medial septum. The channel rhodopsin is also co-expressing a YFP protein. So you can see in green the expression of the opsin. And basically what I did is the mice were running on a circular track, and alternate laps had light on. So every time the mouse crossed this region of the track, the light was either turned on, or then for the next trial, the light was turned off and the mouse would get a reward at this location on the track. And so if you remember, I told you that theta tends to occur between 6 to 9 hertz, generally around 8 hertz. And so we delivered light at different frequencies. Mostly we delivered it at 8, 10, or 12 hertz. And so 10 and 12 hertz are frequencies that are outside the endogenous range of theta, whereas 8 hertz is a frequency that's within the range of theta. And for a large part of the talk, maybe I'll show you the results for either of these frequencies. And if I haven't mentioned anything, then that means that the results were similar across all stimulation frequencies.
So just one question here, Ipshita. Was it necessary to do a stimulation, I mean, or the light at a certain frequency, or could you just leave it on or off, for the entire? What was the necessity to do the light stimulation
I think
at frequency?
We could do on or off, but that would basically completely perturb theta instead of change the frequency. And so one of the questions that we had was, does the frequency itself of the oscillation matter, not just does the oscillation matter? And so to get at that, we had to control it at different frequencies. It also is a little bit harder. For example, if we try to go beyond 20 hertz, it gets extremely hard to control the oscillation, and then the endogenous theta just comes back. So I think if you don't control the cells within a range that they are physiologically capable of doing, the system sort of tries to rebound back to its baseline.
Okay. All
right. Yeah. So over here, what I'll show you is an example trace of what happens to the local field potential when the stimulation turns on. This is the raw LFP, and this is the filtered LFP as the mouse is crossing in this position. And when the red dot becomes blue is when the light turns on. And it's a little bit hard to see, but maybe you can appreciate that instantaneously the frequency of the oscillation shifts to a faster one. And we can also see this effect is persistent for the length of the trial. So here the mouse is running back, the light is still on, shining at 12 hertz. And maybe one thing you can already appreciate is that while the frequency is there, there are bouts when the amplitude of the oscillation is quite low. And then when the light turns off, the oscillation again sort of goes back to its slower endogenous range. And these are reflected in these wavelet spectrograms, where we're basically looking at the power in a certain frequency band within this oscillation. And so you can actually see that there are times when the amplitude of the oscillation is really low, so you can't really estimate clearly the frequency. But there's a shift between the endogenous range to a faster range for example, in this example, 10 hertz and 12 hertz. And so this was the first thing that we really wanted to get at, that is there a dissociation between the amplitude and the frequency of theta oscillations? And in fact, according to past literature, we already know that the cell type we were controlling in the medial septum, the GABAergic cells, they are the ones that control the frequency, but not the amplitude of the oscillation. So the amplitude is controlled by a separate cell type, which are the cholinergic cells in the medial septum. And so you can see this again, that during no stimulation, you've got varying frequencies and varying amplitudes within a certain range. And then during 10 hertz and 12 hertz, the frequency shifts, but the amplitude continues to be unmodulated by the manipulation. And another property of these oscillations is that they actually increase with running speed of an animal. So over here, for example, this plot and this plot, you can see that both the amplitude and the frequency of theta oscillations increase as a function of running speed. And then when we do the manipulation, we lock the frequency, but not the amplitude. And so these two are dissociable within the medial septal circuit, basically.
Sorry to interrupt. Can I ask?
Yeah.
I was wondering if you characterize these medial septum neurons, and like the general functions of medial septum, I believe are, I might be wrong, but I think they're involved in olfactory functioning, and I'm not sure if they're also involved in general arousal states. Could you please comment on what exact neurons you're stimulating?
So I don't know. They may be involved a little bit in the breathing rates or combining breathing rates with other ongoing brain oscillations, but the prominent role of medial septum is thought to be involved with the hippocampus and entorhinal cortex, basically, because most of their projections go to the medial temporal lobe. In fact, if you lesion medial septum, you see memory impairments that are the same as a hippocampal lesion, for example. The medial septum basically has three cell types, three prominent cell types. It has GABAergic cells, the one that we're manipulating. It has cholinergic cells. So the medial septum is part of the basal forebrain, and other parts of the basal forebrain go throughout the rest of the cortex and control arousal and attention and probably the things you were alluding to. The medial septum projects only to the hippocampus, so it controls the cholinergic tone only in the hippocampus and the entorhinal cortex. And then they have a third cell type, which are the glutamatergic cells, which there was a very interesting paper maybe 5 years ago, where they stimulated these glutamatergic cells and found that that induces running. So basically, the more you stimulate the cells, the more the mice start running. So I think it's a combination of combining locomotion, arousal, and sort of setting the pace for the rest of the brain to follow with these external or internal variables.
Just a small follow-up. Did you? Yeah, thanks. It answered my question. But I was wondering if you did any behavioral studies after you stimulated the GABAergic medial septum neurons? Did you find any memory deficits or some changes when you just had these subtle
So
changes?
that's sort of the punchline
Okay,
of the talk. But yeah, that's basically the point. So I mean, we are not the first time reporting that memory impairments are there, but I guess the correlation between oscillations in memory wasn't there, but the correlation between medial septum and memory has been there for a very long time. So we know lesioning medial septum or silencing medial septum causes memory impairments.
Thank you.
Thank you. Okay, so yeah, so the answer for the first part is yeah, we can gain control over these oscillations, and we can specifically quite nicely change the frequency. There have been many, many other studies that have also looked at these the other cell types that I mentioned and their impact on memory, and many of those I don't have the time to go into, but if you're interested, feel free to email me and I can send you details on those papers. So we now can shift the frequency of theta oscillations to a different frequency. So firstly, what happens to spatial firing? And so what I'll show you over here is an example. So this is a 10 Hertz stimulation. And as I said, the stimulation was on every alternate lap. So this is the average of all on stimulation laps, and this is the average of all off stimulation laps. And then this is a lap by lap breakdown. And so it's basically lap 1 and then 2, 3, 4. And what I think you can already appreciate is that, for example, this cell over here fires at a certain location, which is this location, every time the stimulation is on. But every time the stimulation turns off, actually prefers to fire in this other location over here, slightly further down the track. And we saw many examples of this, where, for example, this cell just turns off every time stimulation was turned on. So it stopped firing completely. This cell, a couple of the fields remain the same, but then it developed a new field. And then similarly for this one, there was a new field when the stimulation was turned on. And this is a very common phenomenon in hippocampal place cells. They are known to remap, which basically means they change where they fire according to the environment. And this is a critical feature of them to be able to distinguish between different environments. So if you go from one room to another room, a cell that was active in a certain location will now fire at a completely random place. And the idea is that this allows you to disambiguate between similar situations and still remember the details. And so the question is, what is causing the remapping? Is it due to the change in the frequency, or is it something else? And so there was one very important thing that happens during stimulation, and that is there's a giant blue light that's flashing in front of the mouse's eye. And so the first thing that we wanted to do was control for this light, of course. So we did a separate set of animals where now instead of channel rhodopsin, we express GFP in the medial septum. And in this case now, there's no change in the oscillation. The theta frequency remains the same because we're actually not turning the cells on, we're just artificially turning the light on and doing nothing. But interestingly, we find that the same effect on place cells still happens. So this is one cell where the cell remaps between the on and the off trials, and another cell, for example, where the cell remaps between the on and the off trials. And so basically what that meant is that we were seeing these remapping effects that were happening because of the blue light during stimulation, rather than any of the manipulations that we were doing. And so if I quantify this, I can do this measure called rate map stability, which is basically just all we're doing is we're correlating the on trials and the off trials. So for example, this is a correlation, dividing the stim off laps by half and correlating the two halves. So you see a pretty high correlation. This is the same thing for on. This is stim on versus off. And then this is what you expect from a random shuffle. And so basically what we find is that regardless of stimulation frequency, there's a systematic remapping that happens upon stimulation. And not only is the map less stable during stimulation, but then it drastically is different from the no stimulation map. But what's important is the same effect is observed for all stimulation frequencies. And I won't go into too many details, but I saw very similar things also in the entorhinal cortex. So for example, these are spatial cells in the entorhinal cortex that actually, the interesting thing was there was less remapping observed in the open field compared to the linear track, but both grid cells and place cells in entorhinal cortex were intact more or less, barring small changes during the stimulation. And so the takeaway from this is that spatial firing was altered, but it's independent of the oscillations changing, but rather is dependent on the manipulation itself. And importantly, it's to the same extent across different frequencies, which will become important for the later part of the talk. So all you need to know is that there's some impairment, but it's the same regardless of whatever frequency we stimulated theta at. So now we know what happens to the spatial tuning. How is the temporal properties
So
of these cells?
one question, Ipshita, again is, so it's hard to know, from the animal's perspective, obviously, but do you have any guesses for why this might be? I mean,
you mean
yes, the light is
why?
turning on and off. I mean, at different frequencies, and doesn't matter.
Yeah.
But is this, what I'm trying to get to is, for the animal, is this light turning on, associated with, let's say, a different context, as you were referring to earlier, and then it sort of just remaps the place set.
I think it's a matter of how salient this light is, because a lot of people use optogenetics and they've seen varying effects. I'd say our paper is one of the most striking in terms of the blue light simply being able to cause remapping. We did shine the light really bright, and we should have blackened the ferrule or made sure that the light couldn't be seen by the animal. And I think it is a different context. Like, it's very obvious to the animal that this is now a light on trial versus a light off trial. So maybe
it's like entering a different room, essentially. I mean,
yeah, basically. And we know actually the extent of remapping that we observe is not, so within the hippocampus there's different extents of remapping. If you go to a new room, it's called global remapping, where your map is completely altered, and that would be what you would see by the shuffle correlation levels that I showed. Instead, if you keep the same room but you do very small changes, like you make the box gray instead of black, or you change the floor texture, or you change the shape a little bit, then you see what this thing is called rate remapping or partial remapping. And that's more similar to what we have. So it's the same room, similar context, but slightly different features which are represented.
Okay, great, thanks.
Yeah, so now if you look at how temporal firing is affected by this manipulation, I'm first going to explain, again, like maybe you're familiar with this, but I remember when I first started, I found this hard to understand. So what we'll do is we measure the oscillation frequency of a cell. And I told you that these cells tend to fire at a certain frequency. And the best way to do that is to calculate the autocorrelation of the cell. And so I'm just going to go through exactly how autocorrelation is generated. So what you see here is a spike raster. So every line, imagine, is the timestamp of a single neuron firing. And you can already see visually that this neuron is really rhythmic, because it fires in bursts and then pauses, and then fires in bursts and then pauses. So we can quantify this rhythmicity by basically correlating the cell to itself, but just shifting the timestamp shifting the time series by a little bit progressively. So for example, in this case, we're correlating the cell to itself with a slightly shifted timestamp. And so this correlation is pretty high still. It lies somewhere here. If you shift it more, the correlation drops a little bit. More, it drops really low. But then if a cell is truly rhythmic, then after a certain point, you'll see the correlation rise again. So for example, in this time point, the cell is almost at the same temporal scale as the gaps. And so over here, again, you see a high correlation. And so with this autocorrelation, you can then extract the frequency of this autocorrelation, and it gives you the firing frequency of a cell. And so what we basically did is the same analysis. Over here are two example cells from the medial entorhinal cortex. In gray is the autocorrelation during no stimulation trials. And basically these cells are not very rhythmic. That's what you can see over here. And when we give the stimulation, either 8 hertz or 12 hertz, you can see this drastic shift in the autocorrelation. So now the cell is firing very rhythmically at 8 hertz or at 12 hertz. And we can see the frequency based on the time lag between the first two peaks of the oscillation of the autocorrelation. So the cell strongly now begins to fire at 8 hertz and then at 12 hertz. We can do the same thing in the hippocampus where we see more interesting results. So hippocampal cells tend to be, depending on where in the entorhinal cortex you're recording, most cells aren't that rhythmic or theta rhythmic. Hippocampal cells tend to be very endogenously theta rhythmic. So over here you see a clear theta peak even during no stimulation sessions. And during stimulation, you can kind of shift the peak a little bit, but you don't have as strong control of the cell's oscillation frequency as we did in the entorhinal cortex. And this is better quantified in these heat maps over here. So in this case, the top row and the bottom row are the same cells, but each row within this heat map is a single cell. And the hot colors or the red is basically its preferred firing frequency, and then they're sorted by their preferred frequency. So most cells tend to lie around the theta range, like around 8 hertz during baseline. And you can see during stimulation that these cells are perfectly locked to whatever the stimulation frequency is. So over here it's 8 hertz, then it's 10 hertz, and then it's 12 hertz. And a large fraction of these cells can basically just shift to whatever frequency you're imposing onto the local field potential. In the hippocampus also, this is true, but if you'll notice, especially for these lower frequencies, 8 and 10 hertz, the cells are actually, most of the cells are firing at a frequency, as I mentioned, slightly faster than so the white line is the stimulation frequency, 8, 10, or 12 hertz. And then you can see most of these red, the peak of the cells is to the right of the line, which is also there at 10, but much less so at 12. So there is a frequency dependent effect in the hippocampus as to what frequency they prefer to fire in. And the way I'll quantify this is basically by subtracting the cell's preferred frequency to the local field potential. And so anything that's more than 0 would mean the cell is phase processing or has a frequency that's faster than the LFP, otherwise it'll be at 0. And so you can see this within the medial entorhinal cortex, where the difference, cells are around the frequency, stimulation frequency, or like a little bit faster, but not that much faster. I hope these plots are making sense. So this is the difference plot for 8 is pink, green, blue is 10, and then blue is 12. And you can compare this to the hippocampus, where you actually see this cluster of cells much higher than the 8 hertz line, a little bit higher for the 10, and then almost riding on top, similar to the entorhinal cortex for the 12. And so again, the point we're trying to make is that, especially in the hippocampus, it seems that these two ongoing frequencies are somewhat distinct. And so the oscillation sets the base frequency, and then the intrinsic cellular frequency seems to be set by some different mechanism. But there is a frequency dependent component of it, and there's a limit to how much the cells can accelerate. So maybe beyond 10 hertz, it's really hard for the cell to keep up and keep firing at, say, 13 hertz, for example. And so what we find is that these cells shift their frequency, but to different extents between the hippocampus and entorhinal, and it's in a frequency dependent manner. So importantly, this also seems to suggest that at higher frequencies, the hippocampus and entorhinal cortex are not in tune anymore. And so they're actually probably having more of an impaired synchrony in terms of firing. And so now finally with these results, we can see that do we see any changes in behavior and memory? And so to test this, we used a task that's very frequently used for hippocampal dependent memory. It's basically an alternation task. The mice have to go left, return, then there's a delay area where they pause for however many seconds you impose. And then they need to remember that they had gone left in the last trial, and so they need to go right in the next trial. So they have to alternate. So they need to remember where they came from and then go. And then this is just an example. This is actually not the maze that we used. It's a different maze, but just an example. The mouse gets the reward. The blue over here is actually the light, so you can see how prominent the blue light is. This, for example, is a delay zone, and you'll see now when the mouse runs down, he's going to hesitate a little bit at this choice point. And then he makes this call to go to the other direction. And so basically what we're going to do is quantify the performance on how well they alternated. 50% is chance because there's a 50% chance of getting something right. And so the stimulation was done in blocks of 10 trials. So there were 10 no stimulation trials, then 10 stimulation trials and alternated. We also imposed different amounts of delays. So the task is supposed to be hippocampus dependent only if there's a long delay, but for shorter delays, or especially for no delays, there's no hippocampus component of the task because you don't need to hold anything in working memory for too long. So you can see that during the stimulation, we could control the frequency of oscillation, not during the GFP trials. And basically what we find is for the GFP, there was no effect on behavior. There was no difference from no stim. But during the stimulation trials, there seems to be an effect that is more so for 10 seconds delay, less for 2-second delay. And maybe you can already see over here, but it seems to occur only at frequencies greater than 10 hertz and not at 8 hertz. So we were really intrigued by this, that there's a frequency dependent effect on memory. And so to study this further, we looked at more stimulation frequencies to see that is this really true. And we actually do find that at any other frequency that's below 8 hertz, there's no impairment in the memory. So the memory impairment specifically arises if we try to accelerate the cells beyond the endogenous range, as you can see in these bluish plots. And finally, the question we try to address is there any specific part of the task that's important for like when the oscillations are most important? And what we did is we divided the maze into different sections, and each of these sections are supposed to have different roles for memory. So for example, the return arm is when people think encoding happens, which basically is the formation of the memory. And then the central arm, the stem, is when retrieval happens. So when you're actually trying to remember what you've already generated, the memory that you've already made. And so when we targeted the stimulation to these different parts of the maze, we basically found that the impairment specifically arises when the stimulation is delivered to these return arm trials, suggesting that this oscillation or timing or whatever is important for memory it plays a greater role during the encoding or the generation of the memory rather than the retrieval. And so in summary, what I can show you is that memory is impaired in a frequency dependent manner. And considering all the other effects that I showed you, this strongly does suggest that it's the timing that's important for behavior. And it goes beyond the spatial tuning, which of course is important for behavior. But in this case, it seems to be the timing that's more important. And so to answer if the oscillations play a role, it seems to suggest that they enable temporally precise firing, and this synchronization is required for memory formation. And with that, I'd like to thank my lab members from the Lloyd-Gibb lab, which is where all this work was done. This work was the result of 2 publications, and one of them is a co-first author publication with Claire, who I now realize is actually not in this photograph. And my current lab at NYU. And thank you for your attention, and I'm happy to take questions.